Welcome back, Fungi Fanatics. This time, we're taking a closer look at the bacterial side of things as we culture Pseudomonas fluorescens as our MHB, take a closer look at what MHB is and why it's critical for Amanita Muscaria, and just walk through why they're essential. Let's dive into it. Now let's talk MHB, Mycorrhizal Helper Bacteria. This is our Amanita project. Out in the wild, Amanitas and other mycorrhizal fungi form symbiotic relationship with trees and plants around them. The mycelium will form hyphal cages around these roots and spread out into the environment. And more recent research has suggested there's a third organism in this relationship, MHB. Specifically, what can be found with a lot of these Amanitas is Pseudomonas fluorescens. This bacteria helps facilitate the uptake of nutrients between the mycelium and the tree. This MHB zone is in between the roots themselves and the mycelium. The MHB zone is where the mycelium and the roots are, and that's where you can find the cultures of this bacteria thriving. So that's what it looks like in its natural habitat outdoors. But how can we replicate this indoors? And is MHB one of the missing steps for this? So why are we culturing Pseudomonas fluorescens then? If we can culture this bacteria and then apply it to the root system of our host plants, we can let that colony start to grow. Once we know that that colony is developed within the root system, we can inoculate it with our Amanita muscaria mycelium. The idea is that this MHB will help facilitate the relationship and allow that mycorrhizal connection to form. So hopefully, after we inoculate this with both the MHB and the mycelium, and this is fully colonized, we'll hopefully start to get fruiting. But there's a lot of steps that we have to take before we can even think about that. So what's next for this Amanita project? First of all, it's going to be successfully cultivating our Pseudomonas bacteria. Not only are we going to have to successfully learn how to cultivate it on agar, but we're going to have to figure out a broth solution. So we're able to have a liquid form of this culture to make it easier to inoculate to the host plants. After we introduce our MHB to the host plants, we also have to wait for that to hopefully successfully colonize. And then furthermore, we have to introduce the mycelium and wait for successful colonization there as well. Now, hopefully after all of these steps, we can give them a few weeks to fully colonize the substrate and make sure that they enjoy the environment and that we monitor the pH levels and any other factors that could impact our growth. Hopefully with these steps, we'll be successful. Thanks for tuning in. Now it's time to prepare our beef bouillon agar. We're going to be using about 160 milliliters worth of water. Now we're going to go ahead and measure out 2.5 grams of agar powder. Now into our water will go our 2.5 grams of agar powder, 1 teaspoon of bouillon powder. And now we're going to begin to go ahead and heat our mixture to go ahead and dissolve everything in. We'll bring this to a light boil, let everything combine thoroughly, and then we can pour it into our agar plates and go ahead and do a no-pour sterilization inside of the Instant Pot. With the mixture about as dissolved as I think it's going to get, there's still some floaties. I don't think this bouillon powder is the best choice, but we're going to try it anyway. I'm going to go ahead and pre-pour these to do some no-pour sterilization afterwards. Definitely some chunkiness to them, but it shouldn't be the end of the world. Now these are going to go cool in front of the flow hood until they get wrapped up and pressure cooked. The rest of this I am just going to store so I can put it in the fridge. If this doesn't end up working out, I'll have some more agar ready to go. Now our plates are mostly cooled down. They can be wrapped in aluminum foil. Wrap it however you want. I like to wrap from the bottom so it's flat because the agar will liquefy and re-solidify in the Instant Pot. Make sure it's totally raised on the water. I've raised it about 2 or 3 inches by using a silicone basket and a trivet. Wrapped in aluminum foil. We're good to pressure cook on your favorite setting. I'm going to do this a little higher just because I'm not going to be here for a little bit. I'm just going to let this go a little bit longer to kill any extra contamination that could be there, just to give this the best chance of success. Once this is done, we can let it cool and we can go ahead and move on to inoculating with our bacteria. We'll also be testing our bacteria on our PDA plates to see if that's a viable method for cultivating this bacteria as well. I'll see you in the lab once this is done. Now our plates have finished. Let's go ahead and unwrap these. Take a peek. Alright, these plates, like I said earlier, do have a little bit of floaties in them, but they turned out looking alright. A little bit of condensation, that won't be the end of the world. I went ahead and made myself an inoculation loop with just some really thin gauge wire. Just wrapped it around the little rod and twisted it out. I can heat this with the induction sterilizer, cool it off in the Agar slant, take a little bit of our bacteria culture, and we're going to do some streaking of that bacteria, our Pseudomonas fluorescens. Start with opening our case and taking a look at our slant. Beautiful. You can see the nice streaking on there, a nice zigzag. The Pseudomonas bacteria. So we're going to start with one plate at a time. We're going to be working in quadrants, 1, 2, 3, and 4, doing 4 separate zigzag streaks. Start by sterilizing our loop. Let's take a very small amount. And streak our plate. Beautiful. We're going to sterilize the loop. And now we're good to wrap this dish. Beautiful. And now our first streaked plate is good to go. We're going to sterilize our inoculation loop again and continue to work within 4 to 6 inches of within the laminar flow hood. Just like we did before. And we're going to streak this plate again in quadrants. Clean our loop. And we're good to seal up. Beautiful. And that wraps it up. We tested an experimental beef bouillon powder agar. I'm not really sure if it's going to work or not. It's a little bit floaty and it's definitely not ideal, but it was cheap and something I already had, so we'll see. We've successfully inoculated our Pseudomonas fluorescens, however, to all 3 dishes, 2 of our bacteria agar, and 1 with PDA to test its viability. I'll be monitoring these dishes closely over the upcoming few days to watch for growth, as well as think of ways to make this broth. I also wanted to select some new host plants, transfer our Amanita cultures to T2, and test the viability of that Amanita liquid culture I have to make sure that it's not contaminated. Unfortunately, from the previous video, our white clover died, and I think this was due to exfoliation. The window that it was growing at did not have great direct sunlight, so they grew tall, thin, and ended up falling over because they were too weak to hold themselves up. Let's also take a look at that blue-eyed grass, which is almost dead, but not quite. Now, I did inspect the roots to these earlier, and the roots are healthy, and they're not dry. So I actually believe it's just due to poor sunlight. I'm not very good at growing plants, obviously, yet, but I'm hoping that by keeping these in a little bit better sunlight, moving the window that it's at, hopefully they will recover well. I do want to experiment with some ryegrass though, and maybe look at like a pine bonsai or maybe a very small pine sapling to experiment with next. As always, experimentation is about learning through the process. So stick around as we uncover more about this fascinating symbiosis. Thanks for watching, Fungi Fanatics. I'll see you in the next one.